Hello, everyone. I'm Daisuke Kihara from Biological Sciences and Computer Science. Um, we are working on computational methods development in protein structure and function prediction. So here I will briefly show you some of our work. We develop methods for protein structure modeling for cryo-EM, or we also do the structure prediction from amino acid sequence, 3D structure prediction, or protein-protein docking, protein-drug docking, and also protein function prediction. Now I show you two methods we have recently developed for cryo-EM. Cryo-EM on the left is a powerful method for solving protein structures. One weakness is that sometimes the resolution of the image is so low and needs a computational methods to assist identifying protein structures. So what we are doing is we develop a methods for building a protein structure model from low resolution cryo-EM map. The one method we have developed about 2 years ago is called MAINMAST. What MAINMAST does is, given a cryo-EM map, we want to trace it will trace the C alpha position of the protein by identifying the dense points in the cryo-EM map and then connecting them into a graph. Recently, we further improved our approach so that it can also model complex structures, multi-chain complexes in an EM map. We also use deep neural network, deep learning for many projects. Deep learning has been very successful for processing 2D images, like a 2D image classification. And that can be very suitable for analyzing 3D cryo-EM map. So what our approach does is that given a cryo-EM map, it will scan, our method will scan the cryo-EM map with a small box and then identify or assign the secondary structures of proteins at each box positions. This is some example at each panel A, B, C, D, E, F. On the left side shows the actual proteins 3D structures in the cryo-EM map. And on the right are our assignment. As you can see that, okay, the red dots are corresponding to alpha helices and the yellow dots are identify the beta strands. And as you can see that actually our assignment agrees very well to the actual correct answer. Now we also apply deep learning for protein 3D structure prediction too. So on the left side, this is an example. Left side is a model of a protein. This is a beta lactoglobulin. And on the right, we show the superimposition, actually comparison between our model against the crystal structure, experimentally determined structure, which are in pink. As you can see, they agree fairly well. And then this is done again by deep learning. By deep learning, we predicted distance map of amino acid residue pairs. So this is a recent very exciting work. Now we can predict which residue pairs are interacting, or actually distance between residue pairs from amino acid sequence only by deep learning. So, well, we also do a sequence-based approach work. This is predicting function from amino acid sequence. From amino acid sequence, we are predicting, well, there's a couple of steps, but at the end, we predict the list of functions for this protein. This is very important because when a genome sequence is determined by experiment, most of the proteins do not have function assignment, but our method can predict over 50% of the function over for of over 50% of the proteins in the genome. We are also participating in a genome sequencing project for whale. So if you join our lab for a rotation, we will have at least 2 meetings in a week, group meeting with everyone in the lab, and then individual meeting with you. And during the lab rotations, if you are willing to write a small program, yeah, probably we will do that. Or we could also do some survey on a specific topic, or do some research with existing software. So you don't necessarily need to be able to write a program to do the rotation. If you join our lab as a permanent lab assignment, then you would need to write some program program. But at the very beginning, you don't necessarily need to be able to write a code, because you will learn it in maybe in a few months. So in the current situation, studying in a lab is very difficult. But since our lab are all computational, it is much easier to do the remote work. Actually, most of our students are working at home. A couple of students are coming time to time in the office for some reasons, but most of the students are working at home actually, and still do the meaningful rotation. So please, you are welcome to consider do rotation with us. Well, another thing I wanted to mention here is bioinformatics course I'm teaching this fall semester. It's named Computational for Life Sciences, offered from Computer Science Department, but this is actually not for non-CS students like you. We will cover some bioinformatics methods and also learn elementary Python programming. So hopefully you are interested in taking this course. This is our lab. We have students from both biology and computer science, and several undergrad students, and actually interacting with students from different backgrounds is one of our strengths. Let me know if you have any questions. You are very welcome by contacting me to contact me by email or anything else. Okay, thank you.